In September 2024, a patient with multi-drug resistant Pseudomonas infection received engineered bacteriophages. Within 72 hours, infection cleared. No antibiotics. This frontier of AMR therapeutics. I'm Dr. Anthony Ure Analoka. On this channel, we decode global health through evidence-based analysis, one crisis, one solution at a time. Do click the like button and subscribe to get the YouTube algorithm going. Now let's get into it. This content is educational and is based on World Health Organization 2024 to 2025 data and the CDC reports. It's not medical advice. For antimicrobial prescribing or stewardship implementation, kindly consult qualified healthcare professionals and infectious disease specialists. The necessity of innovation. The discovery drought in conventional antibiotics demand exploration of fundamentally different therapeutic approaches. Novel modalities that do not rely on small molecule antibiotics offer potential to circumvent existing resistance mechanisms while providing treatment options when traditional antibiotics fail. In other words, novel therapeutics bypass resistance mechanisms. And the rationale extends beyond immediate treatment needs. Bacteria evolve resistance to any selective pressure given sufficient time and exposure. Anyone, even novel antibiotics eventually face resistance. Therapeutic diversity, meaning having multiple classes with distinct mechanisms, creates redundancy in our antimicrobial arsenal. When one approach fails, alternatives remain available. The WHO's 2024 research and development priorities for antimicrobial resistance identified several promising therapeutic modalities beyond conventional antibiotics. Bacteriophage therapy employs viruses that infect bacteria. Monoclonal antibodies neutralize bacterial toxins or enhance immune responses. Antimicrobial peptides disrupt bacterial membranes. Microbiome modulation restores protective microbial communities. CRISPR-based approaches target specific resistance genes. Each modality offers distinct advantages and faces unique challenges. None will replace antibiotics entirely. Rather, they complement existing tools, expanding therapeutic options for resistant infections and potentially delaying resistant development through combination therapies targeting multiple bacterial vulnerabilities simultaneously. You see, the challenge lies in translating laboratory promise into clinical reality. There are regulatory pathways designed for small molecule drugs, but these do not readily accommodate living therapeutics like bacteriophages or genetically modified bacteria. Clinical trial designs also struggle with personalized approaches like patient-specific phage cocktails. Manufacturing biologics proves more complex than synthesizing chemical compounds. And there's the issue of reimbursement systems, which provide inadequate incentives for narrow spectrum targeted therapies. Nevertheless, progress accelerates. There are multiple phase 2 and phase 3 clinical trials which test novel antimicrobial modalities. Regulatory agencies develop adaptive frameworks. The evidence base grows. We'll begin with a quote. As the physicist Max Planck observed, science progresses one funeral at a time. In antimicrobial therapeutics, progress requires not deaths but paradigm shifts in how we conceptualize infection treatment. Bacteriophage therapy. Bacteriophages are viruses that infect and kill bacteria. They represent one of nature's most abundant biological entities. An estimated 10 to 30 first phages exist on Earth. They have co-evolved with bacteria for billions of years, developing exquisite specificity for bacterial targets. Phages are viruses that kill bacteria specifically. The therapeutic concept is elegantly simple. Identify the bacterial pathogen causing the infection, select or engineer phages that specifically target that organisms, then administer the phages to the patient. The phages replicate within bacterial cells, lysing them and releasing progeny to infecting neighboring bacteria. In other words, as bacterial load decreases, the phage numbers decline proportionally. Therapy self-regulates. Clinical evidence supports feasibility. The Center for Innovative Phage Applications and Therapeutics at UC San Diego has treated over 100 patients with compassionate use phage therapy since 2018. Published case series document successful treatment of multi-drug resistant infections, including carbapenem-resistant Acinetobacter baumannii, Pseudomonas aeruginosa, and Mycobacterium abscessus. There are many patients who have exhausted all conventional antibiotic options before phage therapy. The September 2024 case that we mentioned in our opening involved a 62-year-old patient with prosthetic joint infection caused by extensively drug-resistant Pseudomonas aeruginosa. Multiple antibiotic regimens failed over the course of 8 months. Now, removing the prosthesis will leave the patient non-ambulatory. Researchers isolated phages from environmental samples, confirmed activity against the patient's bacterial isolate, and administered them intravenously and locally. Within 72 hours, inflammatory markers decreased. At 6 months follow-up, the infection remained cleared and the prosthesis retained. However, there are challenges that complicate wider implementation. Phage therapy requires bacterial isolation and susceptibility testing. These are capabilities that are unavailable in many clinical settings. Regulatory frameworks lack clear pathways for living biological therapeutics. Manufacturing personalized phage preparations proves complex. Host immune responses may neutralize administered phages even before achieving therapeutic effect. Bacteria can develop phage resistance, though often with fitness costs that restore antibiotic susceptibility. There's the Belgian company Affael Bio and the Australian company Amplify Biosciences, and multiple academic centers conduct clinical trials evaluating standardized phage preparations for specific infections. These studies aim to demonstrate efficacy in controlled settings, informing regulatory approvals and clinical guidelines. You see, phage therapy will not replace antibiotics. However, it offers valuable additions to our therapeutic arsenal for resistant infections. And this brings us to the issue of artificial intelligence in drug discovery. AI and machine learning technologies accelerate antibiotic discovery by analyzing chemical space far more efficiently than traditional screening approaches. You see, these computational methods identify promising drug candidates faster and more cost effectively than synthesizing and testing millions of compounds empirically. Notice, AI screens millions of compounds in just hours. In February 2020, researchers at MIT published a landmark study in the journal Cell demonstrating this potential. They trained a neural network on molecules with known antibacterial activity, then screened over 100 million compounds in silico. The algorithm identified a molecule named Halicin with potent activity against multiple drug-resistant bacteria, including Acinetobacter baumannii and Mycobacterium tuberculosis. Importantly, Halicin's mechanism differs from known antibiotics, thus suggesting novel activity unlikely to face existing resistance. Subsequent studies expanded this approach. Researchers at Stanford used machine learning to identify antibiotics effective against slow-growing bacterial persistence, populations that evade conventional antibiotics and contribute to chronic infections. Scientists at the University of Toronto employed AI to predict antibiotic toxicity, helping prioritize candidates less likely to cause adverse effects. Now, the methodology extends beyond discovering entirely new compounds. AI can also optimize existing molecules to improve potency, to reduce toxicity, or to overcome resistance mechanisms. It can predict drug-drug interactions, enabling more effective combination therapies. AI identifies repurposing opportunities, existing approved drugs with previously unrecognized antibacterial activity. And thus, commercial applications emerge. There are companies like In Silico Medicine, Recursion Pharmaceuticals, and Benevolent AI, who apply AI to drug discovery across therapeutic areas, including antimicrobials. The reduced time and cost potentially make antibiotic development more economically attractive, partially addressing market failure issues. However, computational predictions require experimental validation. Here lies the crux of the matter. Many compounds showing in silico promise fail in laboratory testing due to solubility issues, due to poor bioavailability, or due to unanticipated toxicity. Clinical development, in spite of all this, still requires years and substantial investment. AI accelerates discovery but does not eliminate downstream challenges. Nevertheless, computational approaches represent powerful tools. I'll give you a quote from Thomas Edison. As he noted, genius is 1% inspiration and 99% perspiration. Where does AI come in? AI handles much of the perspiration. Now we look at immunotherapies and monoclonal antibodies. Immunotherapeutic approaches harness the human immune system rather than directly killing bacteria. These strategies include things like monoclonal antibodies that neutralize bacterial toxins, antibodies that enhance phagocytosis of bacteria, and vaccines that prevent infections from occurring. In a nutshell, immunotherapy enhances natural defenses. There are several FDA-approved monoclonal antibodies that target bacterial pathogens. Bezlotoxumab prevents recurrent Clostridioides difficile infection by neutralizing toxin B. Obitoxaximab and raxibacumab treats inhalational anthrax by neutralizing anthrax toxin. Now, these demonstrate proof of concept that antibody-based approaches can effectively address bacterial infections. There are other broader applications under investigations. These include antibodies against Staphylococcus aureus surface proteins, Pseudomonas aeruginosa virulence factors, and Klebsiella pneumoniae capsular polysaccharides. Phase 2 and Phase 3 trials evaluate safety and efficacy in various infectious contexts, be them surgical prophylaxis, adjunctive therapy for bloodstream infections, prevention in high-risk populations. Some of the advantages include high specificity, reducing collateral damage to beneficial microbiota, mechanisms distinct from antibiotics avoiding cross-resistance, and potential for long duration of protection with single doses. Some of the challenges include high development and manufacturing costs, intravenous administration requirements, and narrow spectrum limiting use to confirmed pathogens. You see, therapeutic vaccines represent related approaches. Now, unlike prophylactic vaccines that prevent infection, therapeutic vaccines administered during infection boost immune responses against established pathogens. Research programs target Staphylococcus aureus, Pseudomonas aeruginosa, and Mycobacterium tuberculosis. The key insight in all this is that killing bacteria represents only one approach to treating infections. Supporting the immune function to control bacterial growth offers complementary strategies. So we see, how do we combine these approaches and how do we use integrated strategies? The future of antimicrobial therapeutics likely involves integrating multiple modalities. Combination approaches using antibiotics with adjuvants, phages with antibiotics, or immunotherapies with conventional treatments may prove more effective than any single modality alone. Note this, combination therapies target multiple vulnerabilities. The beta-lactamase inhibitors exemplified successful combination therapies. These compounds do not kill bacteria. Rather, they inactivate resistance enzymes, thus restoring antibiotic effectiveness. If we are to expand this concept, researchers investigate adjuvants that enhance antibiotic activity through various mechanisms, such as disrupting efflux pump, permeabilizing bacterial membranes, or inhibiting DNA repair systems bacteria use to survive antibiotic stress. Combining phages with antibiotics shows particular promise. Studies demonstrate synergistic effects where combination therapies prove more effective than either alone. Phages may select for bacterial mutations, reducing antibiotic resistance as trade-offs for phage resistance. Antibiotics may prevent evolution of phage resistance. The combination attacks bacteria through two distinct mechanisms simultaneously. Now, clinical implementation requires careful consideration because not all combinations prove synergistic. Some may even be antagonistic, actually. Sequencing and timing matters. Regulatory pathways for combination products add complexity. However, the biological rationale remains compelling. The World Health Organization emphasizes that no single innovation will solve antimicrobial resistance. Therefore, comprehensive strategies combining stewardship with infection prevention, with access improvement, and with therapeutic innovation prove necessary. Novel therapeutics represent critical components, but not standalone solutions. We'll quote the systems theorist Donella Meadows, who wrote, "The system is a set of things interconnected in such a way that they produce their own pattern of behavior over time. Antimicrobial resistance requires system-level resistance." Researchers and clinicians, which novel therapeutic approaches seem most promising for clinical implementation? Share your perspectives in the comments. So we'll conclude by saying these few things. Number one, bacteriophages curing multiple drug-resistant infections. AI discovering novel therapeutics. Immunotherapies enhancing natural defenses. The therapeutic revolution extends beyond conventional antibiotics. None will solve resistance alone, but together they expand options. Innovation requires research investment, requires regulatory adaptation, and requires clinical implementation. The frontier exists. The question now is whether we will adequately resource exploration. The future of infection treatment demands it. In the next episode, we'll look at some success stories. The Netherlands, Sweden, and Thailand reduce resistance rates while maintaining patient outcomes. It shows that evidence-based solutions work. Once again, do not forget to subscribe to Global Health Decoded at Global Health Decoded on Instagram, TikTok, Facebook, LinkedIn. Next, we'll look at success stories. Did you nail this? Maybe, maybe not, but always fact-checked, peer-reviewed. Dr. Anthony Ure and Nawioka. See you on the next one.